Right, so we'll discuss today why do we need nano sensors. So what are the advantages of nano biosensors? We have seen the field of miniaturization has proved to be beneficial for many different areas. We have looked at microelectronics for devices, energy. Some of us have interest in energy areas, so we have looked at some energy sensor areas as well. But for the biggest reason or biggest application where I can see is Bio-Nano. And there are many benefits, many reasons why we believe that this is having or will have a big impact in nanotechnology, will have big impact in biosensors, not just biosensors, in sensing as well as the therapy, in drugs as well. So reducing the size of biosensor to the micro and nanoscale gives us better signal to noise ratio. It gives the possibility of using smaller volume of samples. If we are talking about smaller sample, we are talking about smaller amount of reagents also. So reducing the overall cost of those sensing platforms, or what we call assays, or as we have seen, the DNA microarray, so microarrays, assays, high throughput screening. So not just that, when we go down, the surface to volume ratio goes down, surface to volume ratio goes up. So we have more surface for the same volume, which means the available sensing area increases. So, and that gives us more opportunity to use same volume, but more surface area, which means more reactivity. And now we make smaller electrodes, we make smaller channels, so we can confine them in smaller spaces and look at the target biomarkers in much higher resolution, if we can call that. So in that case, we can see non-specific binding, and so our overall sensing capabilities are much more selective. So increases the binding efficiency also when we are dealing with smaller volumes, and we have a way of interfacing smaller volumes and smaller biological specimens. Now, the use of devices and use of systems, we have to see there is a distinction, small distinction between bio-nano and nano-bio, but we generally use them in a same sense, or depending on whatever is the application. Now, when we have much more information from the bio side, we can mimic those processes at smaller scale also. So we can come up with newer synthetic materials or natural materials and mix them and try to mimic, and that's an area called biomimetics, where we can learn as things happen in real living system and try to emulate that at small scale, different scales for that matter. Because we have seen many examples where living things have many hierarchies of their size. So micro scale, when you zoom in, you see nanoscale features. So we can create those in this situation. When we talk about sensing, it's sensing and curing. Both have are related to disease. Both are related to diagnosis and therapy as well. Now, when we talk about diagnosis, maybe imaging, it may be the biomarkers, those specific molecules we are looking for, that we want to find the elevated levels of some molecules, or maybe lower levels of some molecules, depending on different diseases have different biomarkers. So higher levels or lower levels of those molecules, which otherwise would be in a normal range, but they go up or down. Now, with that, we can then have a capability of early detection, because we can see small amount and we can see small changes that can come with that. And using nanomaterials like quantum dots and nanoparticles, we can have molecular level detection now. So historically, or as it is, the medicine is we look at organ level, we take some cells out in a biopsy, and now we are talking about looking at single molecular level kind of detection. Now, if you talk about therapy in drug delivery, nanoparticles or gels or matrices where we are using nanoscale features to use self-assembly for our advantage, or do controlled release of drugs or specific site release of drugs, and developing materials that can dissolve and they don't turn into any toxic situation. So that's, and sometimes we want those materials that we use for drug delivery, we want them to be stable for longer period of time at target site. So all these things are which are giving us from Bio-Nano area, we're getting that. So on when we say we learn from the living system, so there's electric circuits, or computing also is something where we are using self-assembly to in areas like molecular electronics or quantum computing, where we want to place small things at specific places so we can have the capability of computing. So, and new materials, fluorescence new materials, which can give us capability to detect and identify changes. So there are many reasons why this field is important and this field is flourishing. And there's various technologies and techniques that are coming up. We have seen some examples already, like Bio-Nano, where DNA and gold nanoparticles are mixed and to do self-assembly. So in some cases, we may take DNA, we synthesize it and use it. In some cases, we may synthesize DNA on the chip itself. So and we can modify biological molecules in many shapes to our advantage. Can you hear me? Okay. Can you all hear me? Okay. All right. Now, there are various ways how we modify DNA, and depending on our surface, whatever chemistry we need, and then we characterize molecules that we put down on the surface. So that's an area what we call self-assembled monolayers and quantification of self-assembled monolayers. So when we make a claim, okay, I have put these molecules, maybe organic or inorganic, let's stick to DNA for that matter. How do we prove that those molecules are there? So there are a bunch of technologies that we use to characterize those things. So one is contact angle measurement. So contact angle measurement is where we use an equipment to see, let me just write down, contact angle measurement, where we put a droplet of water and we see what is the angle of that droplet with respect to the interface. And that contact angle tells us a lot about the surface. It would tell us if the surface was hydrophobic before, and now after doing some functionalization or some treatment, the contact angle has changed. And we have characterized contact angles for many different surfaces and many different functionalizations. So looking at that and comparing results from what people have reported before, let me remove the contact angle word that's written there. So we can use this to show that there has been a change on the surface property, right? So the contact angle is telling us about the boundary condition, the surface boundary condition. And we use an equipment called contact angle goniometer, right? And it's not just limited to the liquid or wet surface. It's equally applicable if we have two liquids or maybe two vapors. So we use that to find out the angle. So if the angle is more than 160, we generally take those surfaces as hydrophobic. Now we put the droplet and we have a microscopic type of thing where we can, or we have a light source which can enhance and give a bigger picture on a screen, and we can take the image, we compare those two. We can look at another technique called ellipsometry. So ellipsometry tells us the change in the polarization of light when it's reflected off a surface. So when you start with the surface, we can take the ellipsometry angle and we get some information from the surface. And once you functionalize it, you do the next treatment, we can see how does the ellipsometry data change, how does the polarization change for that light. So we throw a light, light gets reflected, we measure the light, and that tells us. Another thing we do is XPS, X-ray photoelectron spectroscopy. That tells us elemental composition. So if I put down a molecule, for example, DNA, now DNA brings nitrogen with it, DNA brings phosphate with it, oxygen with it. So now before and after, if I look at the elemental composition, we would see those specific peaks to much more heightened, or those should be absent before, and now we would see that. So that's also a proof of showing that there is chemical changes happen on the surface. Now XPS spectra are known. So we have standard spectra for, or standard data for all elements that could be used, right? So as the name says, XPS, we throw an X-ray and we measure the kinetic energy and the number of electrons that escape from the top, probably 1 to 10 nanometer of the material, and that tells us what is the change in the surface. So in another way how we do is called FTIR, Fourier transform infrared spectroscopy. So what it does is this also identifies and quantifies not just the molecules, but also their vibration mode. So we know vibration mode of different bonds if they are, so the bonds are vibrating in nature. So they would be vibrating like this, if that's orientation, or if they are vibrating on the sideways. So those vibration modes tell us the orientation of the molecules, right? So we direct the radiation in infrared, and we measure the absorption of the radiation, and that tells us the changes in the vibration energy of the molecule. So Raman spectroscopy, another technique, which again, only from the surface, we can measure the change in the angle of the incident energy of the rays. There's another surface plasmon resonance. So we strike a light, some part of the light gets captured on the surface. So that capture would be different from different self-assembled monolayers, or the absence of that, right? So there are a few techniques how we actually characterize our SAM, or self-assembled monolayer, that we have put down on the surface. So now these techniques and these technologies, coupled with smaller devices, smaller dimensions, give us many capabilities. So the nanofabrication is becoming cheap, becoming more accessible, and this gives us capability to make biochips and newer biosensing modalities. So we have now many different SOPs available to us to not just bind the molecules, but to maintain their function, and which is dependent on the structure. So we know now a lot about how we would put things down and how would they behave. So now when we talk about molecular interactions, so that also has given lots of data on what may be the reactions, or what may be the adhesive terminal groups, functional groups that would bind with better between two kinds of things, and like for thiol and gold, right? Sulfhydryl group and gold. Thiol is sulfide, SH group. We know they bind much better, right? So we use these things then in patterning at micro scale, or proteins or DNA molecules, and we then use their binding affinities to our advantage. So nanoscale patterning we have seen in next generation lithography with AFM, with scanning probe, with a lot of things we can create these things at small scale and characterize them, right? So now when we design a system, we have to understand what goes into that design and how we would characterize the maybe sensing or maybe the presence of that reaction. So there are molecular interactions which are done where we immobilize them and maybe we introduce nanoparticles, maybe we introduce quantum dots. So we use the biological specificity to our advantage. Maybe it's antigen and antibodies that we use. The antibody is a protein which is produced by a host to bind to and to activate a foreign particle. So we know their binding. They are made by a host to specifically bind with it. We can take those complexes and put on the surface. So that foreign body is called what we call antigen, right? So it's many times it's a protein. It may not be, but many times it's a protein. So we know those binding between antigens and antibodies. And we know they are very specific. So if we design it well, we would rest assured we would know that the binding is between the antibody and antigen that is of our choice. Now, when we know they'll bind, we modify antigen to carry maybe the drug molecule, maybe another DNA sequence, which is going to do whatever sensing we are going to do. So we modify antigen then to carry and bring whatever we need to bring to study the molecular interactions. Now, there is, of course, the folding structure of our antibody. We need to maintain that. We need to make sure the surface has the suitable hydrophobic or hydrophilic properties so it can help us do the binding. We need to know the pH on the chip is right. So in this design, a lot of things go when we are designing a surface or when we are designing any nanomaterial or using a nanomaterial in that device, right? So pH, the background ionic concentration, the van der Waals forces, electron double layer. So these are the things that you'll have to consider when we are designing a bio-nano interaction. Now, when we nail down all the different ligand receptors, antibody, antigen, DNA molecule, the target and probe DNA molecule interaction, so we put it down. How do we know that binding is occurring? So we do a bunch of things. So we talk about sensors generally, but now we are talking about nanobiosensors. Now we are talking some technologies and techniques that are used to measure those changes that happen on the surface, right? So we used AC measurements, we use DC measurements. So AC would be where we sweep the voltage, sweep the frequency, and we want to see how is that frequency changing. Is there impedance change? Is there, maybe we are looking at the resonant frequency. Is there a shift in resonant frequency? Is there any amplitude that are changing because now you have some binding event that has occurred? DC will be, the simple thing about DC is Ohm's law. Crank the voltage, measure the current, right? If that changes because of the resistance is changing, that would tell us that something is changed in that setup, whatever we're using. Now, redox, again, the reduction and oxidation. So loss of electron is oxidation, right? So it's going to give out an electron. You can measure in on an electrode, or gain of electron would be reduction. So which means again, there is change in potential at the surface. So those can be measured. We use potentiometric devices also where we are measuring potential under equilibrium condition where probably maybe there's no reaction. But then if there are charged species that come along, they would change the interface of the electrode, and we can measure change in potential. We can measure change in voltage, right? So that's now the change in potential will indicate that there is, respect to a reference electrode, that the potential has changed because of some binding event that has occurred, right? So electrical, amperometric biosensors, or potentiometric biosensor, those types of electrical detection. We may look at the change, right? So that's what we call calorimetric biosensors, where we are looking at an image, real image, or we take a snapshot, or we take a video, and then we analyze what is happening and what are the changes in the dye that we have introduced, or luminescence from a bioentity, or quantum dots, or any molecule or any combination which gives out light. So we measure that. Now, in image analysis or calorimetric, many times we are looking at the change in wavelength. So we excite them with one wavelength and we were supposed to get another wavelength. If there is a change, that shift in wavelength can be indicator of our design. So I just put the frequency, phase, amplitude would be for AC. The DC would be more with resistance, with change in resistance, change in the slope, and reduction, oxidation, and right. So these are just, I put some things there. Questions? I think this is slightly more going into how you would design a bio-nano interface, or how we would measure our sensor, right? So how we prove to people that this is actually happening, whatever we put down on a surface. So DNA we have talked about. Let's talk about protein complexes. So antigen antibodies, right? Antigen, foreign thing, antibody that living system produce. So they have inbuilt specificity because antibodies are meant to go and counter them and bind them and neutralize them, right? Because they come from outside world. Now, so we know many of those bindings, but those bindings require that we have the right conditions for that binding to occur, right? And if we don't have the right conditions, so what we mean narrower window condition, which means that all the things need to be taken care of before they actually bind, right? So, and we measure their affinity by, so there is a way how we calculate their affinity. We generally have it less than nanomoles. So they are measured in nanomoles, affinity, binding affinity, and dissociation constant. So these are, you would see Ka or Kd as a measure of the affinity of two molecules, how good they bind with each other. All right, so what do we use? A lot of literature you will see, or the earlier literature was using BSA and avidin. BSA is bovine serum albumin, and albumin and avidin complex. And there is, we call them, they are vitamin. So biotin is vitamin H, or I think it's B7. It's water soluble, and avidin is a glycoprotein, which is found in egg white, and birds and reptiles and amphibians. There's lots of places where it is isolated, and it has 4 identical units, so subunits. So a lot of times we use the BSA to cover the whole surface. Avidin binds to BSA, and then we use biotin, and this orange cylinder thing would be whatever we want to carry with it. Maybe it can be a drug molecule we want to put down. Maybe it is maybe they come with a device, and we want to place device at a specific place. So we would coat a device somewhere else with biotin, and we coat our chip surface with avidin. We let the solution flow. They will find each other because they bind to each other. And once they bind, the whole surface is covered. So protein chips, is the idea is that they would have multiple layers, and then there will be a assay, sandwich thing, what we call sandwich assay. So avidin and biotin are coming together to create the surface chip, whatever functionality we want. So we need to create proper pH for that protein. So that's another interesting point. So we know pH, right? So pH tells us acidic or basic. But when we talk about proteins, for proteins, there is a measure called isoelectric point. So isoelectric point is where the functionality, with respect to their function, they are neutral. And we know what pH needs to be maintained plus or minus for it to be active, right? So which means now we need to adjust our solution to be basic or acidic, because the pI falls somewhere. So pI is what is the pH of a solution where the protein is neutral. So that's another consideration. We have to find the right place. And once we get it down, then we put another solution, what we call blocking agent, which would then cover all the evidence which is not used for binding. So it's just one example of Bio-Nano. We use protein complexes to functionalize our surfaces and to get the functionality what we want. All right, so now there are many issues when we talk about Bio-Nano. Of course, we need to create the right conditions. We need to have the right temperature for right salt concentration. So for example, like for DNA, these things would define that the duplex or double strand structure is stable. You heat it up, they're going to denature, what means that they're going to turn into two single strand. Or if you don't have enough salt, the phosphate backbone is negatively charged, those two molecules will not come together. So we need to provide the positive charge, enough salt so that can shield their backbone and they can come together. So we need to make sure these secondary structures that we wanted those molecules to have, once we put them down, they maintain that structure, because a lot of time for proteins, the binding depends on their secondary structures or their overlap or their folding and all those things which give them functionalities. We need to make sure the surface charges are not strong. So when you're talking about nanoscale, SAM, right? So few molecules thick, the surface, if it's metal, for example, or semiconductor, would have some charge issues where if it's N-type, it has more electrons, it's gonna attract positive ions close to the surface. So we call it electron double layer, EDL. EDL concept is used in many other places also, and especially at nanobio. So this creates an issue where the molecule may not be able to come to the surface because there is strong double layer, strong charges on the surface. We have to make sure these surfaces are compatible with whatever we are using. So there is no reaction between our solution and whatever activity we want to achieve on the surface. So it should be non-reactive for our case or whatever we are trying to achieve with that binding. So, and there's another problem that you create a surface and your biosamples or molecules are in solution. They are roaming around under diffusion. They're diffusing around, right? Brownian motion, random motion. How do you bring them to the surface? So that's where many different techniques or tricks are tried. So to make sure that there's higher probability for that molecule to come and have an interaction with the pro molecule or the surface coating. So we do get the recognition. The orientation of the molecules on the surface, the sensitivity of the sensor, the transduction that we want to achieve, they all go into if these things are not right, so we will not get the right sensitivity. Specificity, I've highlighted that especially for DNA molecules. So DNA has its own features where if you have too much G content, it tends to form secondary structures. Sometimes we engineer them to be there, sometimes we don't want to be there. So delta G here is free energy, Gibbs free energy. So we want to calculate that molecules have enough energy and their melting temperature is high enough that at room temperature, they come together and bind. If their melting temperature is very low, which means even at room temperature, they are going to stay single strand. They will never bind. So the free energy penalty has to be, so we adjust that by the sequence and sequence composition. Sometimes there are mismatches, which will result that the molecules will not be able to bind. So again, for that particular molecule, we calculate. There are tools available or there are models available with which we calculate exactly what kind of sequence we want to develop and how would it come together, and would be, we have realistic delta G penalty with those sequences that we bring together, right? So we use a lot of time we use DNA for self-assembly because DNA molecules by nature, they bind to each other, right? So this is an example of actually where there was a control where there was no capture occurring and a control and the actual surface where polystyrene nanoparticles were captured. So the scheme was made where the probe molecule or the capture probe, what's called here as a thiol group. So it's anchored to the gold on the surface. And now you have polystyrene nanoparticles floating around, and they have another chain which is supposed to be complementary to the capture molecule, to the capture probe. So once they come together, they would bind. Now, in this case, there is this DNA molecule has a biotin, and then the bead has avidin coated to that. So a bunch of specificities that once come together, you see that particles are captured on the surface, and there's no particle capture on the control, where there was possibly maybe there was capture probe was missing, or maybe the biotin target probe molecule was missing. So it depends on your design of experiment, how we do that. So another example, okay, so in this case, the controls may be that we may have a molecule which doesn't have a thiol, which means that they would not bind to the surface and they would get washed away. Another control could be that we have the thiol, but we have non-complementary hybridization. So the molecule that we bring in with the biotin is not complementary, so it would not bind. So that's another control where we would have the actual chip would have the right molecule and the control would have the wrong sequence, same size, wrong sequence. So that would prove that the binding is occurring because there is complementarity in the molecule which is with the biotin. Or there can be another control where we have the beads with no avidin coating. So that can then show, okay, the binding or the capture of bead is happening because we had avidin there. So we have to think through for our experiment design when we are dealing with nanomaterials and especially biomaterials. Now, this binding of DNA has been used for many interesting things. So this is an example where this left picture, if you see, it's 15 microns. So they had these devices which were free floating, and they had the surface chip structure where this device was supposed to go and self-assemble and find its place where it was supposed to go. So the free floating ones had DNA molecules on them. Oh, they had gold on that. So the DNA molecule which is put on the surface had thiol molecule on those. So if you see SH written, that's thiol. Let me highlight what we are talking about here. So in this case, this SH group, which is shown, is supposed to bind to this gold, which is on this free floating device thing. And once it finds it, it gets fixed there. So which means that you could throw a bunch of devices in a chip in a solution, and this can find its place and go and get settled there and connect two electrodes on both sides and become a functional device. So if you have looked at carbon nanotubes or nanowires, we do a lot of these kind of tricks to make sure they fall and find their place where they're supposed to go in your device structure. So another thing which we use here is dielectrophoresis. So dielectrophoresis is a phenomena where if there is a force exerted on a charged body, or especially dielectric particle, where dielectric is where you can have shifting of charges, and that force comes from the different voltage. This would result a movement in that particle or that body that has dielectric properties. It may not be charged, but it would follow the field and follow this non-uniform electric field and follow the force of that non-uniform field and find its place. Now, this dielectrophoresis is a very important property which is used for an important technology called gel electrophoresis. So we have talked about DNA, but how do we actually find a DNA sequence? So gel electrophoresis is the process which we use to find the DNA sequence. So a DNA molecule is charged, right? So a bigger molecule would be more charged than a smaller molecule, but a bigger molecule would have more mass. A smaller molecule would have smaller mass. A gel is a wet polymeric thing which has small pores in it. Agarose gel, which is written here, agarose polymer. So we introduce molecule in that gel and we apply voltage across. What does that do is that results into DNA start to travel under the field. The longer sequence will travel further, the shorter sequence will travel less because it has smaller charge. Now, once we do that, we would get a number of bands from starting point towards the end point. We get DNA would gather. So depending on its length, we would have DNA fragments. They would separate out in different bands. So that separation is a direct result of using voltage across a gel. So let me go back to this slide. So what we do is we separate out different sequences. That's what is called purification. But we also use this to sequence DNA, which would mean that we would cut DNA into smaller fragments. Some would be smaller, some would be larger, depending on if we are doing sequencing or we are doing purification. In purification, we don't want to cleave. We just want to get different lengths of DNA molecules separated out. So once they separate out, then we add a dye, and that tells us the band wherever it appears. So this picture is shown on the right. So we apply voltage with the length of the gel. DNA is negatively charged, right? So it would start traveling towards the positive terminal. And during that passage, once it balances the force out of two, one is negative, one is plus field. Once it balances the forces from both sides, it stops there. So different charge molecule means different length molecule would stop at different distances. And that's what is shown at different steps in this figure. There is kind of what we call it ladder, right? So it's creating a ladder. So from that, we can find out specific sequence, or we can use enzymes to cut our sequence at specific places and then purify DNA for whatever cutting we have done. So we know many restriction many enzymes that are used exactly for recognizing different sequence of nucleotides and then cleave it there, to cut it there. And we use that to let the DNA travel, which has a phosphate backbone, which is negatively charged, and hence it travels in the E field, in the electric field, and charge to mass ratio tells us how far does it go. But mobility, or how far it goes is only a factor of the charge that it carries, right? Which is how long it is, how long is the backbone. All right, so now why are we interested in it? So it is an actual application of electrical engineering, where we use it to separate out different sequences, or to actually sequence an unknown DNA molecule. So we apply molecule, we let the gel run, it results into DNA running to certain sequence, and from that we can find out which one is what, right? So yes, sorry, the longer fragments, longer fragments stay close to the negative terminal and the shorter fragments travel further. We have used this information to make nano devices to actually discriminating between DNA molecules. So we have used this knowledge of DNA backbone charge and how those sequences move in a field. So this is an area called nanopores or ion channels in biology. What we do is we make a very small hole, if that's a nanopore, or ion channels exist. We have studied them. They are on the cellular wall, there are ion channels, or there are nuclear pores, right? So pores, they are basically, we call them, so these are small passages through which specific molecular entities pass. Ions, potassium ions, sodium ions pass. So there are two types of nanopores. So they use the bio ion channels, or we create them ourselves. So what we do is we make very thin membrane and we make a very small hole, 2 nanometer, something like that, because DNA is 2 nanometer wide. And that is used to then measure DNA properties. We can measure with that when a DNA passes through. We can measure with that if there is a specific chain that is passing through. We can measure if there is DNA-DNA interaction going on. We can bind DNA to proteins and look at how the passage of DNA molecules change, what are the forces of binding between them. We have used also to look at biomarkers, proteins that travel through those channels. So here's an example of an actual nanopore. So if you look at this, so there's process. So we start with the bigger hole, then we throw energy at it in a transmission electron microscope, and the nanopore shrinks. So if you look at the lower right picture, we have a nanopore of 2 nanometers. So this open gray area is the opening, and rest is silicon dioxide or silicon, depending on whatever it is. And the left-hand side is showing that as the pore shrinks, the resistance increases. So we start with lower slope. So this is just voltage current, Ohm's law. But as the pore becomes smaller and smaller, the current through that keeps on going down. Resistance keeps on going up, right? So there's an actual picture of, we start with the wafer and then we open trenches, and then we use the focused ion beam to drill a hole, or use the electron transmission electron microscope to shrink a hole in there. And what we get, now we suspend this in the measurement setup and we flow DNA. And when one molecule flows, we are measuring ionic current, we see a dip in current in that current trace. And that tells us a molecule has passed. Many different studies have been done on that. So this is a study which we did many years ago, where we coated a nanopore with specific molecules. So if the molecule was a matched molecule, we could differentiate that from a molecule which was a mismatch, meaning there was a mutation in that. There are many different studies. So if you look at this orange trace, you see spike going down. That one spike is indicative of one DNA molecule going through. Now there are more studies done on that. So DNA is a charged molecule. So when it goes through a nanopore, its charge gets disturbed. So there's lots of studies done that to actually look at the single molecule kind of charge distribution with these kind of devices. So just an introduction on nanobiosensors and why do we use them and what are the benefits of those.